Om Shri Sai Ram. With humility and love, we offer ourselves at Swami's lotus feet. Respected elders and dear brothers, we welcome you all to the National Science Day 2022 celebrations. To start the proceedings, we request our Director Sir and Professor G. Nageshwara Sir to light the ceremonial lamp, followed by the traditional Veda chanting. Sirs.
गणेशाय नमः सरस्वत्यै नमः श्रीगुरुभ्यो नमः हरि ओम् यश्चन्द्रसां वृषभो विश्वरूपः छन्दोभ्योऽध्यमृतात् सम्बभूव समेन्द्रो मेधया स्पृणोतु अमृतस्य देवधारणो भूयासं शरीरं मे विचर्षणं जिह्वा मे मधुमत्तमा कर्णाभ्यां भूरिविश्रुवम् ब्रह्मणः कोशोऽसि मेधया पीतः श्रुतं मे गोपाय मेधा देवी जुषमाना न आगात् विश्वाची भद्रा सुमनस्य माना त्वया दुष्टानुदमाना दुरुक्ता बृहद्वदेम विदधे सुवीरा त्वया दुष्ट ऋषिर्भवति देवि त्वया ब्रह्मा गतश्रीहृतत्वया त्वया दुष्टश्चित्रं विन्दते वसुसानो जुषस्वद्रविणो न मेधे मेधां मयिन्द्रो ददातु मेधां देवी सरस्वती मेधां मे अश्विना उभावादत्तां पुष्करस्वजा अप्सरासु च या मेधा गन्धर्वेषु च यन्मनः दैवी मेधा सरस्वती सामां मेधा सुरभिर्जुषतागु स्वाहा आमां मेधा सुरभिर्विश्वरूपा हिरण्यवर्णा जगती जगम्याम् ऊर्जस्वती पयसा पिन्वमाना सामां मेधा सुप्रतीकाजुषन्ताम् मयि मेधां मयि प्रजां मयि अग्निस्तेजो ददातु मयि मेधां मयि प्रजां मयि इन्द्र इन्द्रियं ददातु मयि मेधां मयि प्रजां मयि सूर्यो भ्राजो ददातु हंस हंसाय विद्महे परमहंसाय धीमहि तन्नो हंसः प्रचोदयात् ओम शांति शांति शांतिः
Om Shri Sai Ram. Now it's my pleasant duty to give the welcome remarks. As you all know, the National Science Day is celebrated on February 28th of every year. This year, the theme for NSD 2022, as envisaged by the Government of India, is Integrated Approach in Science and Technology for Sustainable Future. Can we have the slides? So, the National Science Day on every year is celebrated in the honor of Sir Chandrasekhara Venkat Raman for his Raman effect. The Raman effect was discovered by Sir C.V. Raman in the year 1928. Later, in the year 1930, Sir C.V. Raman is awarded Nobel Prize for the Raman effect. In 1986, the National Council for Science and Technology Communications requested the Government of India to designate February 28th as National Science Day. On this occasion, theme-based science communication activities are carried out throughout the country. Of late, from the last decade, this Science Day is being celebrated in schools, colleges, universities, and other academic, scientific, technical, medical, and research institutions. Various activities, competitions, and eminent scientist talks are being held across India to celebrate this National Science Day. So here are the themes for the last 5 years National Science Day. As we can see, especially the last one, 2021, the theme is apt owing to the pandemic situation. The theme says that it's future of science, technology, and innovations, its impacts on education, skills, and works. Before coming to the National Science Day celebrations at SSSIHL, I want to take few minutes to read out a quotation from our founder chancellor, Bhagwan C. Satsai Baba, where these lines act as a lamppost for the scientific research that is carried out at SSSIHL. I quote, "In the field of science today, there is a great emphasis on research and discovery. But unless the results of the research are applied in practice, it will be an expensive futility. If all the time is spent on research, when is it to find useful application in practice? Nor is there any sense of discrimination in the promotion of research. Those engaged in research seem to be more concerned about boosting their name and fame by their research rather than about promoting the public well-being through the results of their research. Nor do they seem to be bothered about the harmful consequences of their discoveries." The last line actually summarizes the entire paragraph. The scientific knowledge we acquire must be used for the benefit of our fellow men. So societal research has been the prime focus of the research that is being carried out at SSSIHL. So coming to the National Science Day celebrations at SSSIHL, in the initial years, we had talks from eminent scientists and science faculty of SSSIHL on the ongoing research during that academic year. We also had research poster competitions, mainly for the postgraduate students for the research projects they have been undertaking in that year. In 2020, before the pandemic, pre-pandemic era, we had celebrated in a very grand scale NSD 2020. We had multiple competitions such as essay competition, quiz competition, poster competition, and many so were conducted for the Central Trust Vijayagiri schools and neighboring schools from around radius of 10 kilometers of Puttaparthi. We also had science modeling exhibition at SSSIHL Prasanthi Nilayam campus. Here is a news article about the NSD 2020 that we have celebrated in Prasanthi Nilayam campus at SSSIHL. Owing to the pandemic, we could not have a physical celebration happening in 2021, but nevertheless, our spirits were high and our enthusiasm did not die out. So we had NSD 2020 in the virtual 2021 in the virtual mode. We had talks, a week-long program. In fact, we had talks on 24th, 25th, 26th, and also on 28th, 27th February. Owing to this, on behalf of the Science Day celebrations, we had an afternoon session of the entire Science Day with eminent speakers speaking to us on popular science talks. With this brief welcome remarks, here is the program schedule for this year's NSD 2022 celebrations. Here you can see on the screen the morning schedule and the afternoon schedule. So with this, I once again welcome you all to the National Science Day 2020. Now, it's my pleasant duty to request our Vice Chancellor, Professor C.B. Sanjeevi, for the inaugural address. A brief bio about Dr. Sanjeevi. Dr. Sanjeevi has been at the Karolinska Institute, a medical university in Stockholm, Sweden, since 1989. Karolinska Institute is known for selecting and awarding Nobel Prize for Physiology and Medicine, and the institute was established in the year 1810. He heads the Diabetes Immunology Research Group at the Center for Molecular Medicine and is affiliated to the Department of Medicine. His group's focus is on autoimmune diabetes. Dr. Sanjeevi has won several awards, and the most notable one was the Pravasi Bharatiya Samman Award from the Government of India for eminence in one's science field for outstanding work, in this case medicine. It is the highest honor conferred on persons of Indian origin by the Government of India in recognition of their achievement both in India and abroad. Professor Dr. C.B. Sanjeevi assumed charge as the 11th Vice Chancellor of SSSIHL on November 23rd, 2019. Over to Sir for his inaugural address. Professor C.B. Sanjeevi. Thank you. Saira.
Sairam, my humble pranams at the lotus feet of Bhagwan. It gives me a great pleasure to give the inaugural address of the National Science Day being celebrated at SSSIHL. The talk that I will give will be about the Nobel Prize for Medicine and the discovery of insulin, which was made in 1922, and the prize for their discovery was awarded in the year 1923. The Nobel Prize in Medicine, Physiology and Medicine is awarded by the Karolinska Institute in Stockholm, and the selection of the laureates is made by the Nobel Prize Committee functioning at the Nobel Forum inside the Karolinska Institute campus. The Nobel Prize lectures are held in this building on the right, which is a glass building called Aula Medica, where the laureates come and give the talk on 7th of December. So it's the Karolinska Institute that has been selected by the Alfred Nobel in his will to select an award, the Nobel Prize for Physiology and Medicine, since the year 1901. So this is the entrance of the Karolinska Institute, the picture on the left, and you see the building that is immediately to the left of the entrance is the Nobel Forum. So once you enter the Nobel Forum, there is a foyer, and you also have on the first floor of the building an assembly hall where the Nobel Committee meets to decide the prize. A bust of Alfred Nobel is also adorns the Nobel Forum inside the campus. So just before I start to talk about the prize that was given for the discovery of insulin, let me share a few words about Alfred Nobel. Alfred Nobel was born in Stockholm in this particular building, in this site on October 21, 1833. And he's a person whom Stockholmers feel very proud of, or the Swedish people feel very proud of. And he was endowed with the discovery of dynamite. See, you can see the patent that he got in 1863 for the discovery of dynamite, which is still the one that is the most important discovery. During that time, this was such an important discovery, and much of it was important for variety of use of dynamite, which became so famous and also made Alfred Nobel extremely rich. But the discovery of dynamite had led to a lot of conflict situations with the use of the dynamite, which of course made Alfred Nobel very sad and very unhappy. And in his Nobel will, which he was written in 1895, and the will is dated November 27, he clearly states how the constitution of the Nobel Prize and award of the Nobel Prizes have to be made. And this was decided in 1895, 1 year before he passed away in 1896. If you see the award of Physiology of Medicine, what you see here on the left is his will translated into English. And here you see that his award for the Nobel Prize for Physiology of Medicine is one of the 5 awards that he had chosen, the other being physics, chemistry, literature, and peace. And at the Karolinska Institute, the award was initially selected and given by Axel Key, subsequently by Johansson, who was the chair of the Nobel Prize Committee between 1918 and 1926. And the secretary of the Nobel Prize Committee was Göran Liljestrand, who had been serving as the committee secretary since 1918 to 1960. So during this time only, the award of the Nobel Prize for the discovery of insulin was made, and it was quite a controversial decision. So in the will, one of the points that clearly states is in no case may a prize be awarded to more than 3 persons at a time. So this is an important part of the Nobel's will, and since its inception, no more than 3 persons have been selected for the award. And if you look at the physiology and medicine, which is the important discovery that has been identified for the prize, you will realize that the implication of what it says is the preceding year. The prize has to be given for a discovery that has been done in the previous year or the preceding year. This is a very difficult choice to make because in 1 year's time, it is pretty hard to evaluate and identify the discovery and award the prize. Even though this was a difficult choice to make about awarding in the preceding year, but what the Nobel Committee decided is the implication of this particular wording. It is the time between the nomination and the award of the prize is often and correctly considerable, which means even in the previous years, or not just the year, they were able to take it into consideration for the award of the prize. But whatever said and done, of all the prizes that have been given for the physiology of medicine since 1901, there is only one prize that really qualifies for the exact wording as it is mentioned in the Alfred Nobel's will, which is the preceding year. And that is the prize that has been awarded in 1923 to Banting and MacLeod for the discovery of insulin, which they did the previous year in 1922 and had the paper published during that year. So this is the only prize that really qualifies for the award of Nobel Prize. So here is the prize that was awarded for the discovery of insulin, and this particular saga of the discovery even became a book written by Michael Bliss, which is a very famous book on the discovery of insulin, and that had really made an impact in terms of should they deserve it, should they get it. So Frederick Grant Banting and John James Richard MacLeod was awarded the Nobel Prize for Physiology and Medicine for the discovery of insulin. The discovery was published in 1992. The nomination and the prize was given in the year 1923. The choice of the combination was debated for a long time, and Banting, who felt that the prize should also include one of his colleagues, who is actually his student, PhD student, Charles Best, should also have been included in the award. So he felt slated, and in this, what he had done is whatever prize money he got, he shared it with Charles Best. Whatever may be the prize money that he might have shared, but the glory of having been awarded the Nobel Prize was never actually given to Charles Best. So this is the award of the Nobel Diploma that was given to Frederick Banting and John MacLeod. And this is the credit that he had received, which is half the price. And on the left, you can see a picture of the office and the clinic where Frederick Banting worked in Toronto. And he shared half the price. And if you were to visit the house where Banting did much of his work and lived there, this is the house, and there is an eternal flame outside the house that have been created as a flame of hope. He also got featured in 1923 in the cover of the Time magazine, which was a famous magazine even during that time. And outside the house stands a statue of Banting, where you can see him standing and reading at a book. How did he win the prize? Within 1 year of discovery. So that is a very interesting story, which I was able to study from the archives of the Nobel Prize for Physiology of Medicine, which was published during the 100 years of Nobel Prize, in the journal Diabetology in the September issue. So the important character here is a gentleman by name August Krug, a Danish person who actually became very well known as a biomedical scientist for his discovery and series of works on absorption of oxygen and elimination of carbon dioxide in the lungs that takes place by diffusion and by diffusion alone. Until then, this particular piece of information was not known to the world. And he was awarded the Nobel Prize for this discovery in the year 1920. And the publication of this had taken place in the preceding years. So August Krug became very famous after he won the Nobel Prize for Physiology of Medicine for his discovery. And what happened subsequently was that in 1921, August Krug's wife developed diabetes. And he was looking for a ways by which his wife could be cured or treated for her condition. At that time, the lifespan of a diabetic is very short. Maybe they could live only for a few years. So he was at the same time invited to America to give a lecture in Northern America, and in 1922 he visited Boston. So when he visited Boston, he had the opportunity to share a dining table, and the person who was sitting next to him was a very famous person called Elliot Jocelyn. So during the dinner conversation, August Krug was talking to Elliot Johnson about his wife's condition, because Elliot Johnson is one of the famous diabetes doctor and the scientist, and we now have a famous hospital called Joslin Diabetes Center. So Jocelyn and August Krug spoke about August Krug's wife, Mary Krug. At that time, Jocelyn informed August Krug that there is somebody in Toronto who had made a discovery of insulin that brings the blood sugar down. And this information August Krug considered very, very important. And he changed his plan to go to Toronto from Boston. So when he went to Toronto, he was able to see all the work that was done by Banting. And of course, the whole place was headed by John McLeod. So he visited the place as a guest of John McLeod. And he looked through all the protocol and he was very, very impressed. In the meantime, not only that he was talking about his wife's condition, but he thought it was such a great discovery. He went and got an agreement signed, a license for the production and purification of insulin. And he brought this license back with him to Denmark. And he met his personal physician, who was Hagedorn. And Hagedorn was the father or the founder of the current Novo Nordisk, which is producing insulin and making it available to the people in the world. So Hagedorn was the person who took the license and made it into a commercial production for insulin. And August Krug was associated with Hagedorn for quite some time. And that became the Nordisk Insulin Foundation, which is now the Novo Nordisk. So what is interesting here is that the insulin that they produced in Denmark helped keep the blood sugar of August Krug's wife under control. And when August Krug's wife died in 1943 from cancer, nobody knew that she had diabetes, but she died of cancer. And everybody thought that diabetes never even knew to any of them. So this is how the discovery was made into a product and product brought to the market. So that became very interesting. If not his wife would have gone the diabetes, he would not have made the visit to the Toronto, and the Banting and McLeod would not have been awarded the Nobel Prize for medicine. So it is very unusual for someone to receive the Nobel Prize for Physiology of Medicine the same year as he was nominated. It had never been the case. So as can be seen from the archive material from the Nobel Assembly of the Karolinska Institute, from which I had some access, it was true that Banting and McLeod deserved the prize. And why did they deserve the prize? They published their work on the discovery of insulin in 1922 and were nominated for the first time in 1923. And Banting, Kreil, Benedict, Krug, McLeod, Stewart, August Krug, this was the publication. And Charles Best, on the other hand, was never nominated. Even though everybody felt that Best should be awarded the prize, he was never nominated at that time. Only nominated candidates could be considered for the prize. So in his nomination letter, August Krug, as a Nobel laureate, when you make the nomination, such a nomination takes a lot of weightage with the Nobel Prize Committee. So he wrote like this: with the information which I personally have obtained in Toronto, which also, although less clearly so, emerges from the published work, one may conclude that the credit for the idea behind the work which led to the discovery undoubtedly goes to Banting, which is a young, apparently well-talented man. However, he would definitely not have been able to carry out the investigation, which from the start and during all stages have been supervised by Professor MacLeod. So in his nomination, he clearly gives credit to John MacLeod as an important person in the discovery, along with Banting. So Charles Best was never mentioned, so he was never included in the prize. A letter from the nominee, August Krug, to the Secretary of the Nobel Prize Committee, Joran Liljestrand, dated 1923, which is on the year when he nominated him, says that I have on the left hand side reproduced the picture of that letter. As you understand from my discourse, it is my opinion that the discovery of insulin is of extraordinary both theoretical and practical importance, and it will hardly surprise you that I intend to submit a nomination that the Nobel Prize be awarded to Banting and Professor MacLeod. So this was his nomination letter that he wrote to the Secretary of the Nobel Prize Committee. But few years later, this particular award had created such a big controversy because it left out a lot of other people who could have also won the prize. So August Krug, few years later, he writes to the Secretary of the Nobel Committee saying, I understand that the prize to Banting and MacLeod, ignoring other co-workers, has not been met with absolute consent on the other side of the Atlantic Ocean, and that especially Banting is offended by the fact that Best was not included. However, I am convinced that the correct choice was made. So this letter clearly shows the influence of August Krug on the Nobel Committee in terms of deciding the prize for Banting and MacLeod. So this is one of the important communications that tilted the balance in favor of Banting and MacLeod to receive the 1923 prize. So if you look at the questions that have been asked, why was MacLeod included? And why was not Charles Best but also James Collip excluded? So Bliss is an author who wrote this book in 1982, and he had done a thorough analysis and arrived at the conclusion that the choice was made by the best possible. Others are of the opinion that Nicholas Paulescu, Joseph von Mering, Oskar Minkowski would have been perhaps worthier laureates for the prize. So this was the opinion shared by Rolf Luft of Karolinska Institute, who was also chairman of the Nobel Prize Committee, and he had looked at several of the nominations, including Charles Best, that had happened in the subsequent years. So Rolf Luft is being quoted by Bliss in his book. So some additional information that might shed some light into the situation can be obtained from the Nobel archives. The Paulescu was never nominated. Collip and Best were nominated, but not until 1928 and 1950, respectively. So Best was never nominated until 1950. Von Mering was nominated, but only in 1902 and 1906, while Minkowski was nominated in 1902, 1906, 1912 and 1914, as well as in 24 and 25, but Minkowski didn't get the prize for his discovery. Thus, according to the statutes of the Nobel Foundation, none of these candidates could have received the prize in 1923 because they were never nominated for the prize for that year. So perhaps the choice made was the best possible, or it would have been wiser if the Nobel Committee at that time had explored the situation in greater depth, rather than proposing to the medical faculty of the Karolinska Institute that Banting and MacLeod be awarded the prize at such an early stage of the discovery. So it all boils down to one important fact, that August Krug visited America, met with Jocelyn, and visited Toronto subsequently, and if he had not had those meetings, he would not have nominated. Probably little later, few more people would have nominated Banting and MacLeod, with few more candidates that could have included Charles Best also. See, you can look at the turn of events that at the end ended in favor of Banting and MacLeod for the award of the prize. Now, all these things I have mentioned in my article in Diabetology in 2001. So, the prize for Nobel Prize, which was founded by Alfred Nobel, is of course given for the greatest benefit of mankind. An insulin discovery by Banting and MacLeod was, of course, the greatest benefit to the mankind, as it has been proven even now for patients suffering from type 1 diabetes. There is no other choice but insulin that could be given to them. So this is the prize awarding ceremony for the first Nobel Prize, which was done in Stockholm. This is just a very old picture in black and white. And the most important thing that we have to learn from the Nobel Prize is it is about the new knowledge that we are adding. Societies that have invested in research-based knowledge have prospered. To achieve such a knowledge, it should not be looked upon as a cost, but as an investment. So universities that pursue new knowledge is a great investment for the future. If you don't like research-based knowledge, of course, one can try ignorance and nobody will benefit from the ignorance. You can see with this old man walking in Stockholm, with so many books, at this age, he still wants to gather new knowledge and progress with whatever contribution he can make. So with this, I thank you very much for giving me the chance to talk. I hope this was an interesting talk that would motivate the young minds and the students about what they can do and how the discovery could be made. But the chance is much more important, and at Puttaparthi, we will call it more as a divine intervention for the successes that you may want to have in life. Thank you very much. Sai
Inaugural address and the wonderful talk. Now, it's my pleasure and privilege to introduce the speaker for the next session, Dr. Vinay Kumar Nandakuri. Dr. Vinay Kumar Nandakuri is the director of the CSR Center for Cellular and Molecular Biology, popularly known as CCMB, located at Hyderabad. He is a well-known molecular biologist and has been a scientist at the DBT National Institute of Immunology, New Delhi. Dr. Nandakuri's research interest extensively spans molecular signaling networks in Mycobacterium tuberculosis, the microorganism that causes the TB. His research has found national and international relevance and recognition. His education and research training have been in the Indian Institute of Technology, Mumbai, Indian Institute of Science, Bangalore, Texas A&M University, and University of Virginia, located at USC. He holds a prestigious J.C. Bose Fellowship. He is an elected fellow of the Indian Academy of Sciences, Indian National Science Academy, and National Academy of Sciences, India. Today, Sir will be speaking to us on the topic evolution of resistance in pathogens. Over to Dr. Vinay Kumar Nandakuri, sir. Thank you, sir, for joining us.
Thank you for the.
Sir, I think you're muted, sir. Can you unmute?
I'm not. I think. Can you hear me? Can you hear me?
Yes,
No?
we can hear you.
Yeah. Give me a minute. Can you see the slides? Okay. I would like to thank Dr. Sanjeev. 4 or 5 people there. So today I will be talking about the work done by one of these girls like Sabanas, who's I'm just now indicating. Everything that I'm going to talk about is the work done by her, and I'll give an introduction. See, we do know for the past 2 years, we've been facing a pandemic. The pandemic that basically has taken approximately 30 lakh people. I mean, it has killed approximately 30 lakh people. Even if we estimate even if we assume that the estimates are wrong, there's still around 50 lakh people may have died of this pandemic. That's a big number. And that has all happened in 2 years. But one has to realize, in addition to the pandemic that has been taking us for such a hard ride in the last 2 years, there are other bacterial pathogens, antimicrobial resistant infections, and even the things such as cancer and diabetes and other things. They continue to kill humans. Tuberculosis. Can you please mute your microphones? All of you, thank you. So, tuberculosis is actually one of those. Not my microphone, thank you. So tuberculosis is one of those pathogens, even today, kills approximately 15 lakh people yearly all through the world. And if you really consider the fact that tuberculosis as a pathogen came way back in all the way back to mummies, it is found in Egyptian mummies and all, you will realize that it is most likely the biggest infectious disease that is ever stepped on in the world stage. It kills more people. It has killed more people than anything else, right? So Mycobacterium tuberculosis as a pathogen is an ancient pathogen. That's what I was saying. It still kills 15 lakh people every year. And this is actually become more extensive or more problematic because of the emergence of multi-drug resistance and extensive drug resistance tuberculosis. And India has, unfortunately, the highest burden of tuberculosis in the world, with an estimated 2 million cases annually. That is approximately 1/5 of the world incidence. To give a comparison, we have had approximately 140 million cases of COVID.
Aram sir is on mute, sir.
I'm not muting, somebody else is. It'll be great if they don't. What I'm basically still stating is that it is a pathogen that needs to be worked on, and there are multiple other pathogens that need to be worked on. Why? The biggest problem in most of these cases is the emergence of drug resistance. Let me give you a brief background of how tuberculosis infection happens and what happens during the infection. Transmission is aerosolic. It is taken up by your innate immune cells, and then it starts replicating there. As a response, your host, our body start walling it off with other immune cells, T cells, innate lymphocytes, that is lymphocytes, neutrophils and all, generating something called a granuloma. This granuloma is where the infected cell is in the middle. It is the growth is contained, and it is some kind of walled off by the other immune cells, and which is easy to be detected in what we call as any kind of an X-ray. So this kind of a granuloma has 3 different fates. Your immune cells are very, very proactive and very good. You will completely eradicate the tuberculosis, in which case it is called calcified granuloma. In some percent, around 5% to 10% cases, it so happens that you lose the battle with the pathogen, and pathogen gets out of the granuloma and starts spreading to the other parts of the lung, and sometimes even to the other parts of the body. And the third one is basically the scenario that happens in most cases, where it is a contained infection where we have what I call it as a ceasefire. There is a very clear line of control where the pathogen is inside and host cells are outside. This is all good till something happens like compromised immunity. Where do you get compromised immunity? Sometimes you have, say, arthritis issues, and then you start taking anti-inflammatories because arthritis is an inflammatory response. When you take anti-inflammatory, you can actually activate something like this. Or one has an HIV infection where the CD4 T cells are eliminated. So all these things lead to activation of TB, and that's when it becomes an active disease all over again. As a lab, we work on various aspects of it. We work on signaling, we work on peptidoglycan synthesis, transcription, cell division, and host-directed therapy. But for today, I will focus only on evolution of drug resistance. And the reason is I believe that it is a very relevant topic, considering how all of you have been seeing the viral evolution, and you wonder why viruses keep on evolving. I'll tell you, it's one sense a natural process, but we can talk about it. As just a brief overview on what my lab works on, we actually work heavily on the signaling. Signaling is nothing more than when an external stimuli, no matter what, hits any cell of ours, we get a response. That's signaling process. In Mycobacterium tuberculosis or in bacteria, mostly what we know as a signaling is two-component signaling with the histidine receptor kinase, where the histidine gets phosphorylated, which phosphorylates histidine to aspartic acid residues in the response regulators, thereby giving you a certain response for the system. So in organism like E. coli and many other pathogens have quite a lot of signalings that is mediated by two components. They have anywhere between 20 to 60 to 80 two-component signaling for different responses. And in case of mammalian, we mostly use eukaryotic, then we use protein kinases, and such kinases also exist in bacteria. And that's why they're called eukaryotic-like serine-threonine protein kinases. In these cases, what happens is that the signaling is through phosphorylation of serine, threonine, or tyrosine residues downstream. MTB has 11 such kinases, and 2 of them are essential for its survival, remaining are not. Anyway, I'm not going to focus much on it. We worked a lot on these kinases for a while, and we know that as a kinases actually participate in multiple cellular process in Mycobacterium tuberculosis. Now, that's the background of our overview of the work that we do. What I would like to talk about is drugs and drug resistance. So if you go to a hospital with tuberculosis, what they do is they give you what you call as a DOTS therapy. Generally speaking, when you actually reach the hospital, what you have is what we call as drug-sensitive tuberculosis most of the time. And the medicines that are given to you are isoniazid, rifampicin, ethambutol, pyrazinamide in different combinations. Okay, so the isoniazid, rifampicin, the ethambutol, and pyrazinamide, all these drugs were discovered by 1970. So it's older stuff. It is nothing new. And so when we give these drugs, what happens is, generally speaking, what are the symptoms of tuberculosis? You feel very weak. You actually feel that your appetite is gone. And there are multiple other things. Most importantly, the fatigue levels are very high. People cough blood. Lots of things happen. But when you start taking these drugs, just in about 2 months' time, you feel that you are completely recovered. And that happens. And there lies the problem, because now people do not take these drugs as they are supposed to take as per the prescribed format. Okay, so that leads to compliance issues. Compliance issues lead to resistance. So why does that happen? Every time in our body a genome is duplicated by DNA polymerases, you incorporate certain errors. These errors are directly related to the processivity of the polymerase that is there. So what is the processivity of polymerase? How many errors does it make when it crosses around like 1 in 10 power 8, 1 in 10 power 9? So our genome is approximately 4 into 10 power 9 or 1 point, I don't recall exactly. They are 4 into 10 power 9, something like equivalent to that. So when we are replicating it, if you are making 1 in 10 power 9, you still make 4 errors. So What I'm saying is there are errors, and these errors happen constantly when we replicate. Over and above, many carcinogens or many things, they generally tend to mutate our genomic DNA. And what we have is a secondary response where something called a DNA repair enzymes come into play. What do repair enzymes do? Repair enzymes remove the base that has been mutated and repair the whole process and eventually make sure that your genome is stable and clear. However, mutation is a known fact, and some of these mutations also happen to be beneficial to us, and that's what evolution comes into play. That's how we evolve to become better. And some of them are not beneficial. So what is a beneficial mutant? Any mutation that actually provides an advantage to survive in an environment can be considered beneficial. Any mutation that does not provide that advantage is not beneficial. So as a selection goes, you will always select for things which are beneficial for us, I mean for our body. In the context, bacteria also has the same issues and same problems. What it does is that it also selects for mutations that are beneficial for it to survive when you are providing them or when you are treating them with antibiotics. So if you have mutations that would allow you to become resistant to two of these antibiotics, rifampicin and isoniazid, it is equivalent to multi-drug resistant bacteria. If you have mutations for two of the first line antibiotics and one or more of the second line antibiotics, it is called extremely drug resistant pathogen. And when you have mutations that would allow you to survive against any of the antibiotics, it's called totally drug resistance. Unfortunately, all three things happen. So what are the mechanisms by which resistance can occur? For example, isoniazid that we take as a drug is given as a prodrug. It goes into the body, and at that point of time, CATG cleaves it to make it an active metabolite. And this active metabolite then inhibits an enzyme called INHA. So now, if you make mutation either in the CATG or you make a mutation either or in INH, you will end up getting resistance to isoniazid. And this is actually what happens in most cases. The other drug that we take regularly is called rifampicin. Rifampicin inhibits RNA polymerase. Now, at this point of time, if you are actually going to make mutations in the RPOB gene, which is the beta subunit, these mutations will make it resistant to this particular drug, rifampicin. Thereby, you get resistance to this drug. In addition to that, then you have other ones like gyrase, target oxofloxacin targets gyrase. If that particular target site is mutated, you will end up getting resistance to ofloxacin. So in addition to that, we have something called efflux pumps and efflux pumps. Basically, these are the pumps that are involved in pushing out the antibiotic from within the cell. Means bacteria keeps on pumping it out. So you put in the antibiotics go in, and if it effectively decreases the antibiotic concentration in the cell, you will end up getting resistance. So, and there is in an addition way to generate resistance, this is basically, you basically modify the antibiotic itself. So that the antibiotic that is put in is now certain groups like methyl group and certain things are added so that it does not anymore bind to its target. So there's huge number of mechanisms bacteria can use to modify or to actually generate or to get the drug resistance in the context of this. So in case of tuberculosis, drug susceptible tuberculosis is there 83%, which is where you start out. But non-compliance and the MDR-TB is 17%. And out of that, 54% are MDR-TB, 30% are XDR-TB, which are extremely drug resistant. And MDR-TB cases in India is pretty much high. It contributes to 27% of the MDR cases in the world. So what you need to understand here is the following. Okay, once you have MDR-TB, what would you do? You still need to be treated, right? So earlier treatment with DOTS therapy is anywhere between 6 to 9 months. Now, if once you become drug resistant, once you end up getting drug resistant tuberculosis, the treatment goes up from 6 to 9 months to something like 16, I mean, almost 1.5 years. So, and now the second line antibiotics are given, bedaquiline is given, some of these other things are given. They're all toxic. they're quite toxic to your body, and you end up actually having serious side effects where this treatment is really long and people become extremely weak because of the antibiotics that are taken, and they also affect your other organelles. So better is always to have compliance. If people give you antibiotics, say for example, you go to strep throat or something else to a doctor and they give you antibiotics for 5 days. And what we do, we take antibiotics for 2 days, 3 days. After 2 or 3 days, then you will say, okay, now I am feeling pretty very good and the antibiotics stay in your house, but you don't take. All these things are the reason why we are actually increasing the drug resistant bacteria in the world. And there are the only problem with this is there are only one or two ways to overcome drug resistance. One is either some way tweak with it, generate new antibiotics, or completely discover new antibiotics. In both cases, we end up actually getting, we have to actually go for discovery. The problem with this is any antibiotic that you want to do or discover takes approximately 10-year pipeline, even if a new antibiotic is discovered, for it to come into market. And then you have a lot of toxicity studies and all. So it's a long road, the discovery and evolution. So it is important that we actually take care of this aspect. And this is my sincere request to all of you to complete your therapies that are given. So there are mechanisms in addition to what I have told, and that is what one needs to understand. That is what I am going to talk about the work today. And this is all done by one girl. Her name is Sabha Naz, in collaboration with my PhD mentor, Umesh Varshne, and Paritosh from DU South Campus, Sidra Khan, Priya from my lab, and Yogendra Singh from DU. So what we have done is very simple. We have taken existing available genome sequences that were present or deposited in the central database by people. Means we have not done anything. What they have done is the following. They have basically taken clinical samples, and the clinical samples were then cultured. DNA was isolated. Whole genome of the whole sequence of the bacterial genome has been identified. And what we do at that point of time is that basically we are having all the genome sequence analysis, genome sequences available. They are segregated into either monodrug resistance, or MDR, XDR, and all those things. So we had approximately 2,300 drug-sensitive strains, sequences from drug-sensitive strains, and another 400 odd sequences from drug-resistant strains. So we looked for 2,773 clinical strains, out of which drug susceptible, sensitive, and we made it into 3 parts. And then we look for the genome, we look for the variations that are only found in drug resistant bacteria, but are not found in drug sensitive bacteria. So these variants, after doing all kinds of statistics, came out to be 188 variants. That can be divided into 4 parts: as synonymous variants, non-synonymous variants, upstream gene variants, stop frame shift mutations. I will elaborate a bit on all 4 of them. So essentially, whenever you have a mutation compared to the reference genome, which is found in one particular strain, it's called a variant. Okay, if the variant is found in a statistically good number of strains, then it is a relevant variant. Okay, so synonymous variants are those variations which do not change the amino acid sequence. Non-synonymous variants are those which change the amino acid sequence. Upstream gene variants are those in which the variations happen in the promoter regions, thereby creating an issue in terms of the transcriptional the efficacy. The stop frame shift mutations are for if you have a change in the frame, which essentially you don't get a functional protein. After doing all these things, we ended up getting those with which contribute negatively and which contribute positively. But we were looking at those which are found in MDRs, not found in the sensitive strains. And then we looked into synonymous mutations, where the amino acid is not changing, but the mutation is such the the codon is changing. What we notice is that the codon usage actually changed drastically when you make that single change. So why does it matter? In our body, we have tRNAs that will actually translate, or that will take care of looking at the codons. So some of these things, the tRNAs are less, and they're called rare codons. What is happening in this case is that we observe that our MDR XDR strains are pushing them towards rare codons. You can very clearly see that rare codons are lower in our original laboratory strain. In MDR XDR strain, it is much more. Why is that relevant? Which increases the translational efficiency, thereby decreases the amount of protein that is present, and that may be contributing to XDR, the drug resistant phenotype. The other one is non-synonymous mutations, which is what we were interested in. As expected, we got a lot of genes which are already known to be involved, or the mutations in these genes are known to be involved in evolution of the drug resistance. We got lots of them. Okay, so when we got, like, for example, leucine 452 prol in RNA polymerase RPOB gene makes it resistant to rifampicin. CatG, serine 315 threonine makes it resistant to isoniazid. So we ended up getting this. This is kind of expected. The methodology you are coming up with is correct. We also got a lot of mutations in transporters, which is what I was talking about. Efflux pumps and these kind of things would decrease the intracellular concentration of the drug. So these are the things which we identified in the 188 that we did in our study. But what struck is that we identified mutations in DNA repair genes. Now, a while ago, I was telling you, whenever you have a mistake in your genetic material, it needs to be repaired. Like if your car is not having is having starter issues, it needs to be repaired. So the repair is done by repair enzymes. They remove the bases that are wrong and correct them with a certain process. So these repair genes that we've identified was actually a surprise to us, because we thought DNA repair is a central important mechanism for the survival. So basically what we identified is also that these DNA repair gene mutations are found in conjunction with the other drug resistant mutations that are identified, like isoniazid or rifampicin. Because our original selection was that, it is not a surprise. Anyway, so what we did then looked at it is, what is the role of DNA repair genes in increasing the drug resistance in this pathogen? See, whatever we are talking about is very specifically from Mycobacterium tuberculosis. That does not mean such a or similar or many other mechanisms are not present in other bacteria. One of them is mutation that we identified was in a base excision repair enzyme. The second one was nucleotide excision repair pathway. And the third one, homologous recombination pathway. If you're not a biologist, you may be wondering what they are. Bottom line, they are there to correct the mistakes that happen in our genomic DNA due to multiple reasons. So our hypothesis was simple. If you do not have any mutations in any DNA repair genes, spontaneously, due to many reasons, you end up getting some mutations. And one or two of these mutations in millions and millions of bacteria ends up being advantageous for the survival in the host, and eventually making it MDR or XDR TB. When you have a mutation in a DNA repair gene mutation leads to higher rates of mutations in these bacteria. Higher rates of mutations, some of them would be resistant to the antibiotic because you're putting the stress. If you're putting the selection, you will always go with that direction, eventually evolving drug resistance faster. This was the argument that we had, and we needed to test it. So what is that we actually identified? We identified mutations in five genes. We tested on two. For today, I will only focus on one. MUT1 is a DNA repair enzyme. Whenever you have a mutation from G to become atoxG, if you don't repair it, will become GC to AT. So what you do is that you take out this base and then you repair the process using MUT1. So what is that we identified is that we identified a mutation in certain part of this gene which changed the amino acid. So this change may be good, beneficial, may be disadvantageous. To find out that, we needed to generate a mutation in the Mycobacterium tuberculosis genome. Means you take out the original gene and then you can test it whether the mutant that you have, whether the change that you have identified in your analysis, is it irrelevant or irrelevant. So we generated what we call as a gene replacement mutant, commonly known as knockout. When you knock out, a gene of a particular interest is taken out and replaced with an antibiotic cassette, as you can see very clearly. So such things are called gene replacement mutants or knockouts, or simply called mutants for the sake of our purpose. So then what we did is we took the wild type, we took the Mycobacterium tuberculosis mutant, we performed something called mutation rate analysis. When you perform mutation rate analysis, you're looking at the evolution of mutations when you put an antibiotic stress. As expected, normally here it goes the other way. 10 power minus 9 is lesser mutation, 10 power minus 8 is more mutations. So when you take S37RV, this is smegmatis in this case, smegmatis strain, native strain, you get certain number of mutations, 1 in 10 power minus 9. But when you actually take out the DNA repair gene, the number of mutations increase, right? So the number of mutations increase. And interestingly, what you do is then you put back a functional copy back into the gene. When you put a functional copy back into the strain, that number of mutations are coming down, which indicates that whatever increase that you've seen is because of taking out this particular gene. And what we did then is we identified a certain mutation in the only drug resistant strains. We put that back. And what happens is great. It is basically it's basically showing that it is not anymore functional. So same thing has been done for the UVRB. So what I'm essentially trying to say is that the mutation that we identified by performing such an analysis told us that in certain drug resistant strains, there is a mutation that has happened due to whatever reasons, which is making this particular enzyme non-functional. If this is non-functional, it should have higher abilities to evolve. To test this process, we have done an infection experiment where we either gave a stress or not gave a stress for 72 hours during the infection. So what is this experiment? Experiment is simple. You take a tuberculosis strain, and what is the host for tuberculosis strain? Macrophages. So we take macrophages from the mice and we take these strains and we add them to that. After the infection, we allow it to happen for around 120 hours. Now we take out the strain that was there. So what we did is we gave sometimes an rifampicin stress, some other antibiotic, ciprofloxacin, or total 3 different antibiotics, isoniazid stress. And what we did is after 120 hours, we took it out, we grew them, did this again, did this again, did this again. Repeatedly, when we do, what we call is an evolution happens. Something that advantageous for the bacteria gets selected. So we eventually looked into the ability of this evolved strain to survive in the presence or absence of antibiotic or whatever. So here is that in the absence of antibiotic in itself, the pathogen had a higher ability to survive when you give the just go through the repeated infections. When you complement it, it comes down. When you go back, I mean, when you put the mutant, it is as good as not complemented. The same thing was with INH and rifampicin, ciprofloxacin. The interesting part is you end up getting higher and higher and higher number of mutations. So we did then a next experiment where we actually infected the guinea pigs and looked into what happens to the survival of the pathogen when you have mutations in the DNA repair gene. To cut the story short, when guinea pigs are infected in 56 days later, when we look at it, what we find is that it has accumulated more mutations when the DNA repair gene is taken out, or we don't know whether it is accumulated, but it has a better survival advantage. And when you put it back wild type, it comes down, but not the mutant. So this happens in this thing and spleen and lungs. So finally, we did an interesting experiment where we competed one strain, which is wild type, with the mutant, or mutant which is complemented with the wild type, mutant that is complemented with the mutant. They're competed against each other. In this case, if the mutant had an advantage, it will show you higher CFUs relatively compared to the other. So when we did this experiment, total CFUs were same, but here is the bottom line. What you see is that you get much higher levels of mutation when higher levels of the mutant strain surviving compared to the wild type, because it has a survival advantage. But when you complement it, that advantage is lost. When you complement it with the mutant, again, it has an advantage. So bottom line that we are trying to tell you is, resistance in bacteria comes due to various reasons. And some of the reasons, we have identified a new reason in which resistance can evolve faster, which is essentially DNA repair gene mutations. And these things help in bacteria to evolve resistance faster. This is something that is currently in circulation. Hopefully, we'll be able to publish this work very soon. But what I'm essentially trying to say is that evolution of drug resistance in bacteria is a common phenomenon, and it is not an easy question to us, easy thing to solve if we actually do not actively take care of this by making sure that we do not use we do not abuse the antibiotic regimen that is given to us. Please take the drugs as they are prescribed. Otherwise, you are contributing to the evolution of drug resistance in bacteria. And that ultimately, there is no way any discovery rate can keep up with this particular problem. As long as our immunities are good, well and good, eventually, otherwise, this will be one of the other major killers that will continue to happen. Even common bacterial infections can actually give you very bad outcomes. Thank you.
Thank you, sir. Thank you, sir, for the wonderful lecture on the tuberculosis.
You're
and,
muted.
Are we unmuted?
Yes.
Thank you, sir,
Thank you, sir,
for the wonderful lecture
on
TB
TB and the drug resistance and how to handle the thing, and also the research insights that you have given regarding the work that is happening in your labs. Thank you once again, sir, for taking out time from your busy schedule and giving this lecture. So I request the audience, if they have any questions, they can unmute their mic and ask Vinay, sir. Over to the audience. Any questions do we have? Kindly introduce yourself and then ask questions. Do we have any questions?
I see one or two in the chat box.
Yes, sir. I request you to kindly respond, sir, for one or two questions in the chat box.
So, for example, there is a question from Shivaratnakar, which is, have you performed whole genome sequence analysis after evolution of the repair gene mutant and compared it with the reference genome? This is actually the question we got from the referee, and we knew we have to do this even before we send the paper. But nevertheless, we did not do it because it costs a lot. So now we are going to do it. We have no option but to do it. We are working on doing this experiment, but our prior experience is that if you actually mutate a DNA repair gene, which we have done this earlier, but we have done it not from the evolution point of view or not from as an experiment. Okay, and that time we noticed that if you don't have a DNA repair gene, you get, we have performed WGS, whole genome sequencing, and we do get higher mutations depending on the selection pressure. If it is ciprofloxacin, we get in the gyrase. If it is rifampicin, we get it in the RPOB. This happens, but we have to prove it any which way.
Yes, any other questions?
Sir, also the fitness mechanisms, like the fitness of the bacterium can reduce after DNA repair mutants, no, sir. So WGS also helps us get an info on that.
So DNA repair, it's like this, okay? Your point is well taken. If you are making a mutation in DNA repair gene, won't it kill the bacteria also? I agree it does. But what happens is you are actually, the bacteria is under stress, the stress coming from the host, correct? So there is stress to survive, sometimes oxygen radicals, nitrogen radicals, and if you are putting antibiotic. So there is a certain level of fitness cost anytime you may accumulate mutations, and Omicron is a very good example of it, right? But that sometimes gets compensated by the fact that it can evade. It can evade your host response. Thereby, you actually can get you can end up actually accumulating more mutations. So it's a compromise strains make. There is a fitness cost, but we don't find a major fitness cost when we grow in regular conditions for these strains. In fact, it seems to be I have advantage when we grow into the host, when we go into the host. How do you? Yeah, tell me.
I have a question, sir. Sir, how do you score a variant with regard to its tolerance or intolerance?
See, what we do is we continuously grow the variant, and after a particular point of time, we put antibiotic selection on it. Okay. So on a plate, we look at the number of colonies you get at a particular dilution in the wild type and in the mutant or in the complemented strains, and you score by the numbers, and then you back calculate with the formula that I have showed. So it's an experiment that is done as it is suggested or it is standardized in other labs.
I am asking about, like, you said that you have downloaded publicly available databases, and then
So
there.
Yeah, you're allowing the comparisons to happen using programs, and you're looking for variations that are found in the multi-drug resistance, but particularly it has to be statistically significant. It is not exactly my cup of tea, and nor do I understand how the variants are scored by the algorithms. But there are algorithms that are already available, which will tell you that a certain variant is statistically significant. Means variants are found any which way. So you can find one variant, you can find multiple variants, but not everything is found in consistently in a certain number of strains. Only then such a variant is considered important. But those things are all essentially statistics, and analysis is done by the algorithms that are already available.
Thank you, sir. I think there are no more questions.
There is something
Thank you,
about
sir.
COVID.
Yes,
There
sir.
is
Yes, sir.
something about COVID. See, I have doubts, sir. People who got COVID after its cure also are they hit by the side effects? Okay, so this is a COVID question. I would take it simply because I think people are looking at Omicron and previous Delta infections. The reason why people who already are infected by Delta are getting infected by Omicron is because there are mutations in the antibody binding region of the spike protein. Those mutations do not allow the binding, and that's why they are able to escape. Would they have severe conditions? They are not as severe as if you have never been exposed to the virus. If you are previously exposed to the virus, either in the form of as in the form of a previous infection or due to vaccination, you do have a higher levels of protection and lower levels of what you call as side effects.
Sir,
Thank you, audience. I'm sorry I have to interject for the wonderful talk, and I request the audience to contact Vinay sir via email for any further questions. We once again thank you, sir, and we are keen and look forward for your research advice at SSSIHL. Thank you once again, sir, for joining us, for taking you from time out from your busy schedule. Thank you, sir. Thank you. Yes. Welcome back. So we now have presentations on various research activities over the last academic year done by the science departments. We'll be having presentations in this order. First, we'll be having Department of Biosciences presentation, followed by Department of Chemistry, Department of Mathematics, Computer Science, and Department of Physics. Over to the science faculty to take the session further. Saira.
Om Sri Sai Ram. At the outset, offering my humble obeisances at Bhagwan's lotus feet, respected teachers, students, and the audience that have connected over YouTube. Saram to everybody. So today, I stand before you with the privilege to present the research findings by the Department of Biosciences as part of the National Science Day proceedings. Next slide. Department of Biosciences at Prasanthi Nilayam campus offers MSc in Biosciences and doctoral research programs with a vision to impart ideal education and shape students as ideal citizens and leaders dedicated to the service of society. And we have the mission of excellent training in fundamentals of biosciences coupled with suitable practical experience and project work. Next slide. Following the footsteps of Bhagwan Sri Satya Sai Baba, as he proclaims, science should serve the society and all research should ultimately lead to societal benefit. In these lines, if you look at the quest of man, it's always in tryst with nature, and tryst with nature is constant in man's quest for knowledge. In this regard, biosciences, we being closest to the nature, studying life and life forms, our research revolves around the endemic and the exotic, taking endemic resources to exotic platforms or bringing exotic solutions to the endemic problems and exotic problems. In these lines, our thrust areas of research are under health and environment and medicinally active natural products. We have 10 research scholars combined and supervised by 5 research supervisors. Moving forward, under the theme Health and Environment, we have Disease Biology Group, Infection Biology Group, Microbial Fuel Cells for Wastewater Treatment and Regenerative Medicine and Tissue Engineering. Under the theme of Medicinally Active Natural Products, we have research being carried out on fauna, on structural and functional characterization of type 3 redox proteins, and natural compounds that are isolated from flora and fungi. By the Disease Biology Group, which focuses mainly on metabolomics in disease systems biology, and they aim and they analyze clinical samples and then isolate and then analyze metabolomics to find out early detection biomarkers and also the markers that are underlying the mechanism of the onset of disease. In these lines, Disease Biology Group studies avascular necrosis of femoral head, rheumatoid arthritis, which is an autoimmune disease, and glaucoma, disease of eye, and multiple sclerosis, so this is a neurodegenerative disease, and Huntington's disease, which are carried out by Disease Biology Group headed by Dr. S. Venkatesh. Then, as part of Antimicrobial Resistance Laboratory, Infection Biology headed by Dr. Pradeep, which focuses on the heteroresistance, genetic makeup of rare pathogens, nitrofurantoin resistance and outbreak analysis. So one of the research focus of AMR lab is to develop prediction models using artificial intelligence and machine learning tools, and mainly focusing on genomic analysis of multi-drug resistant bacterial pathogens, so which was very nicely dealt by Dr. Vinay earlier. In these similar lines, so AMR lab has been successful in attracting grants from ICMR and BIRAC. So in addition to the scientific rigor that goes on in AMR lab, we have artistic skills that come out every year. And we have Dr. Balram Kamari, who is currently working as a postdoctoral fellow, has been awarded second prize for his agar art. Coming to the water lab, which focuses on wastewater analysis, treatment and reuse, specifically working with the sewage treatment plant in Prasanthi Nilayam township as model system. Also, water lab focuses on in silico studies which aim to enhance biofilm formation for biotechnological applications. And also, in addition, bioelectrochemical systems, designing and developing screening and platform for components of microbial fuel cells and so on. And in addition, as they focus on the understanding of various plant products as coagulants of sewage, so they also focus on ethnobotany, survey-based exploratory studies on medicinal plants used in South India, home remedies and folk medicines, and also application of plants in eastern Himalayan regions. Then coming to the protein structure and functional lab, which focuses on type 3 copper proteins, specifically focusing on hemocyanin, which is the component of the blue blood of animal kingdom, focusing on arthropods and also the mollusca. So looking at its characterization, structural and functional characterization. Functionally, eliciting phenol oxidase activity in hemocyanin is also focused upon with structural aspects and the underlying mechanism of its activation, with an aim to devise a protein-based phenol biosensor to detect medicinally and environmentally important phenols. In addition, computer-aided drug discovery is also carried out using techniques like molecular docking and molecular dynamic simulations in order to identify potential drugs from the plant bioactive compounds. One such recent study was to look at their potential to inhibit SARS-CoV-related proteins. Another focus is to look at the protein nanoparticle interactions, where recent study has seen looking at C60 fullerenes interacting with protein like hemocyanin. The department in the last 5 years has attracted grants to the tune of 6 crores Indian rupees. So the granting agencies range from DST, DBT, UGC, ICMR, BIRAC, and certain industrial partners. Department of Biosciences has been seeing progress in the publications, number of publications that come yearly. There has been a steady progress and always beating the older records. Some of the popular journals in which the articles are published have been Scientific Reports, Biotechnology Letters, Journal of Experimental Biology, Phytotherapy, Research, Journal of Biomolecular Structure and Dynamics. Department of Biosciences has many national and international collaborators. And also, in the vicinity, Department of Biosciences within the institute collaborates actively with Department of Chemistry, Department of Mathematics and Computer Science, Department of Food and Nutrition, Department of Physics, and also within department, all the areas that I just mentioned, they generally always work in collaboration and share the ideas in order to bring out better science. With this short presentation, I thank you all for your patient listening. Jai Sai Ram.
and characterization of various organic compounds with potential biological activities such as anti-cancer, anti-malarial, antifungal, anti-inflammatory, and anti-neurodegenerative. His collaborators are IIT Madras, National Cancer Institute USA, ICGEB New Delhi, and VIT Vellore. I specialize in the area of physical chemistry and material science, and my research interests are in the field of computational material science and fluorescence spectroscopy. My team is currently working on the following projects: designing molecules for organic solar cells and OLED applications using a combination of steady state and time resolved absorption and fluorescence spectroscopic techniques. Identification and characterization of secondary metabolites using liquid chromatography mass spectrometry. Studying properties of polymer nanocomposites using molecular dynamics simulation and using this knowledge for designing materials. Flashpoint estimation of complex volatile organic mixtures involving 50 to 200 compounds to be used in perfume industry. My collaborators include ORNL USA, IIT Madras, SRM Chennai, and Takasago Singapore. Dr. Shantanu Roy is an inorganic specialist, and his research group is currently working on the synthesis of biocompatible smart materials. These are characterized by various state-of-the-art techniques. These smart materials could be used as artificial bones or fillers and have potential applications in orthopedics. These are processed into various forms and their performance is evaluated by various biological assays. Dr. Kumar Sai Smaran specializes in electrochemistry and is currently working on energy storage devices. His research focuses on the synthesis, characterization, and fabrication of graphene-based electrode materials to be used in batteries and supercapacitors. They also work on understanding the surface phenomena of electrode and electrolyte interface. Dr. Prafulla Chandra's research group focuses on the application of liquid chromatography in atmospheric chemistry, natural products, and medicine. Currently, he is working on qualitative and quantitative analysis of secondary metabolites like polyphenols, terpenoids, and vitamins in natural products. Identification of biomarkers for metabolic disorders in the human body. Identification of chemical markers which act as cloud condensation nuclei in rainwater. Screening of emerging contaminants present in different water bodies. Dr. Nagasai Vishweshwar and his group is currently working on the development of medical diagnostics using novel nanomaterials, microfluidics, and custom designed devices. Clinical sampling, clinical testing, and device fabrication and testing are also being carried out for applications such as perinatal depression, cardiac monitoring, neonatal jaundice, dengue, and COVID-19. Recently, they have developed a COVID-19 rapid test kit, which was unveiled during last year's convocation. They also have received funding from various external funding agencies such as DST, DRDO, ICMR, and Tata Trust. Thank you.
Salam everyone. I am Sai Shyam on behalf of the Department of Mathematics and Computer Science. Today the focus of my presentation is about the various fields of research that are being carried out currently in the department. I am not going to talk about various fields that have been done because that's the past, and we are right now trying to talk about what's happening so that each one of you, whoever is listening, gets interested in possibly taking up these areas of research. So at the core of our department, the mathematics is like the sun which sustains various other fields which revolve around it. Possibly I could even replace these planets with various fields of science itself and keep mathematics at the center, and I wouldn't be wrong for that. But then for now I will just go ahead with our department. So like you can see over here, the various fields are mathematical modeling, differential equations, computer vision, actuarial science, blockchain technologies, deep learning, data science, and high performance computing. So beginning with the first area, study of differential equations which model certain physical phenomena. Here you see an example of how when somebody searches for oil or mineral exploration, the waves that get created at the surface of the earth and which get propagated down below, these mathematical equations explore to study the refractive index that is caused by these waves and try to understand what is the surge going on for, which is deep below, which cannot be seen very easily. Another example of modeling a real life phenomena is the transonic phenomena, when you have objects traveling at the speed of sound or sometimes faster than the speed of sound. The waves that get created, that particular phenomena gets modeled by equations, and the instability that is caused in the object due to this travel can be an issue. So this mathematical formula helps to eventually design these objects in such a way that they remain stable when the supersonic phenomena happens. Another application is automatically detect movement from a video. Yes, go ahead. So if you look at this, you can see the three squares and you can see a set of lines moving. Now if I were to leave you to guess, to tell me which particular direction these lines are moving, you'll be surprised to know that they are moving in three different directions actually. But when you look at it only from the perspective of the hole, like a pigeon hole, you will only see them moving in one direction, whereas all three of them actually moving in different directions. So this particular phenomena could again be studied using mathematics, and like say for example over here, you're using computer technology to detect the movement of this person. So if there's some way that you can automatically detect it, that's one field of study that's being pursued in the department. But the application of this eventually, the interest for the group that is doing this is, yes, go on to the next slide, is the interest of movement in medical images where blood flow in vessels and various parts of the body can be studied. So this is an application of that aspect of detection of movement. Then another field that the department is actively pursuing research is mathematical ecology and epidemiology. So like you can see, the predator-prey model is extensively being studied, and the three models that are right now being studied by the research group in this particular area are holding type 3 predator-prey model where additional food for the predator is provided, and also time optimal control problems. Yes, move ahead. Then the other area is epidemiology, where mathematical models are being prepared for diseases like COVID-19, dengue, and leprosy. That's the current disease that the department is focusing on. This could go further more. Then complex manifolds is an area, I'm sure each one of you who knows a little bit of science would have heard of theory of relativity. So modeling the theory of relativity that Einstein gave using manifolds is a very active field of research, and complex manifolds are trying to the study that is being performed by this group is using complex manifolds. They want to understand this concept of the Einstein manifolds. So the effects of certain curvature conditions on complex manifolds is being studied by this group. And one more application, which is the image processing application, is automatically detecting diseases from images. So for example, first data is acquired, then the data is represented in a form that could be studied, by the computer and the algorithms. Then they classify and analyze this particular data. Now this particular project is actually a collaboration with the Sri Sathya Sai Institute of Higher Medical Sciences, Whitefield, and is being pursued currently. Another field which is the buzzword today is deep learning. Everybody wants to know something or the other about deep learning. How deeply we know is a different issue, but everybody wants to know something about it. So here you can see that the first area of research, equivalent deep learning. For example, actually saying if you train a network to recognize a cat, when you give it the second image, which is the inverted, it will not recognize it as a cat. So equivalent deep learning is about building neural networks that behave predictably for such transformations. Then if you see the other problem that's being studied is part to whole learning. So what happens is if the neural network is trained to recognize a face and then you give it the different parts of the face, it should tell you that it is not a face, right? If you put the eyes below, the mouth above and all those kind of, it should be able to tell you that it is not a face. So study of such neural networks for recognition of objects is also being studied. And the other thing is, suppose you look at the third problem where question answering, there is a relation between the two spheres and the two cubes, but they are two different objects. So relationships can exist between different objects also. The study to the ability to recognize relationships in neural networks is another field of study that's being pursued. All these have a direct application. And apart from mathematics and computer science, another area that is being pursued actively in the department is actuarial science. For those of us who want to make money, this is the field you need to study more. So the applications that are being studied in actuarial science are plenty. Right now it's a little difficult font for me also to read, so I'll just try to tell some of the problems that are being given. Data-driven fraud detection and prevention model using actuarial and data science techniques. So we all know about fraud detection that we all experience some way or the other in our life, sometimes credit card, sometimes it can just be a pickpocket which is happening. For that actuarial science can't help you, but then if it is a credit card problem, definitely they will be there to help you out. Framework for measuring protection of policyholders' interest in industry level. Sometime or the other in life, you will invest in an insurance policy. If at all you have issues that time, you can contact our department. And yes, if you read more, there are plenty more areas. So at the end, we are trying to get into various fields like medical, actuarial, which is a day-to-day usage of people, epidemiology, and also image processing, trying to make a combination of man and an artificial intelligence overall to make the world a better place. Thank you. I hope I was able to pique your interest. If at all you want to know more about the department's achievements regarding publications and more, the annual report is available on the website. Please, you can consult that. Thank you, Sarah.
Om Shri Sai Ram. Offering my humble salutations at Bhagwan's lotus feet. On behalf of all my colleagues from the Department of Physics in all the three campuses, I'm here to give you a brief summary of the research activities that are being carried out in the department. Research in the Department of Physics has been going on three major areas like photonics, materials science, and nuclear physics over the last 3 to 4 decades. All our research has been in focus with respect to the thrust areas of research identified by the Government of India on energy sector, health sector, and environmental sector, because that's where the government is also funding and science could really be of great use in these areas. I will not go into
lot of details, but I will quickly give highlights of some of the important results and research work which we have carried out over the last couple of years. The first one is the lab on chip platform for biomedical sensing. This work is aiming at creating a multifunction lab on chip device which is compact and which could be integrated with optical and electrical elements. The technique that is being used is called as femtosecond laser micromachining, which is the facility which we have in the CRIF A0 lab. We were able to successfully demonstrate making of these kind of LOC platforms on fused silica, which could be used for biomedical applications. Safer methods of fabrication has been demonstrated. Instead of using hydrofluoric acid, we have used KOH, which is much safer to handle, and truly rectangular buried microchannels have been created use of record lengths and record time, which has been published. And we were also successfully able to integrate the SCRS, surface enhanced Raman spectroscopy substrates, onto microfluidic channels by laser sintering of silver nanoparticles. The next area of work is on design and development of mode locked femtosecond fiber lasers, wherein we aim to produce or make nano, pico and femtosecond fiber lasers and study novel materials that could be used for mode locking and create these pulsed lasers. As of now, we have been successfully able to design and study and analyze the laser pulses that are propagating in these optical fibers, and we were also able to study the pulse dynamics within the optical fiber. This area of work involves a lot of nonlinear optics and concepts related to nonlinear optics. A lot of fundamental physics has evolved out of it. We are looking at development of different types of pulses like solitons, dissipative solitons, etc. And we are also doing some mathematical modeling to study these ultra short pulses that are propagating in these kind of novel laser cavities that we have designed, as you can see in this picture. And this has exciting applications in various fields like supercontinuum generation, micromachining, imaging, and also sensing. The next area in the next work in the area of photonics is the low-cost tomography imaging for middle ear diagnostics. This is another very exciting research that is being carried out over the last few years. The aim is in the development of ABC OCT, affordable B-scan camera-based optical coherence tomography, which aims to provide an in vivo real-time cross-sectional images of human eardrum and bones in the middle ear. The primary concern is to address the concerns related to the chronic otitis media and otosclerosis. As of now, we have successfully demonstrated the proof of principle and low-cost parallel line field spectral domain OCT. We have used innovative time averaging techniques based on Bessel functions, and the development of protocols to maximize dynamic range and signal to noise ratio. We were able to sense very high frequency sub-nanometer vibrations with real-time profilometry of multiple inner layers. So this work will have a lot of applications in diagnosis of ear problems. We have also developed some open source software that could be used to implement these protocols. This work is being it is carried out in collaboration with the Super Specialty and SSSIHL General Hospital. Coming to the area of material science, solid electrolytes, the battery technology which could power the future. The aim is to design and develop a solid electrolyte material for lithium-ion batteries, which could have very high energy density and fast recharge capabilities. The team has successfully synthesized novel materials that has very high ionic conductivity at room temperature, wide electrochemical stability window, low activation energy that could help in migration of the ions easily. These novel materials that has been synthesized and studied are found to have very good physical and chemical compatibility with the anode and cathode materials of the electrochemical cell, and they have very good stability over temperature, and they are chemically inert, and they are also low cost materials. So this could be a very good potential material that could replace the existing electrolyte materials in the lithium ion batteries, and that could lead to very efficient battery materials. Another very interesting area of work in the material science is nanomaterials for remediation of contamination from water. The primary aim is to design and develop efficient defluoridating material for real-time groundwater remediation. Stability of the material, negligible leaching of metal ions, and good regeneration capacity should be the aim of such a material. And the team has been able to successfully synthesize and demonstrate Syria incorporated trititanate nanotube composites, which have very high absorption capacity of 65 milligrams per gram, with very good ultrafast kinetics of 90% fluoride removal in 1 minute, and they were able to successfully defluoridate the groundwater to WHO permissible limits without altering the pH. This material is a very attractive and potential nanomaterial for designing a real-time fluoride water filters for households. This research has a lot of relevance to the Anantapur district by, of course, now Satyasai district, but in general the Rayalaseema area, which has very high fluorinated water. So we are hoping that we would be able to come out with cheap or I would say low cost household water filters that could be used by villages for having safe drinking water. Next please. Another area of research in material science is nanocomposite films for electromagnetic shielding applications, where we are aiming to synthesize nanoparticles with excellent magnetic and conducting properties and develop lightweight, cost-effective and flexible polymer-based nanocomposite films for various applications. The team has been able to successfully fabricate novel tin ferrite materials and composites of such material in PVDF films, wherein these fabricated composite films were showing more than 99% of absorption or shielding from microwave radiation. They are meeting the required industrial standard by exhibiting a total shielding of greater than 30 decibels, and the electromagnetic shields effectively suppresses the majority of radiations by absorption. So this particular film or the material that has been made or fabricated is one of the superior shielding materials as of now. Dense lead-free glasses for radiation shielding applications is another area of research, wherein we are aiming at developing lead-free, environmental friendly shielding materials for gamma radiation. The team has been successfully able to produce lead-free glasses of binary bismuth borate systems, which has very high density comparable to lead-based materials. The gamma radiation shielding properties have been investigated thoroughly, and a good research paper has been published. And these transparent dense glasses have shown better shielding properties, and they are promising materials that could replace lead-based glass materials. A very exciting and very important area of work which is going on in the department under the broad area of nuclear physics, or I would say applied nuclear physics, is the multimodality intraoperative handheld gamma probe. In the last couple of years, we have already successfully demonstrated that from a room size state-of-the-art gamma camera, we can come down to a portable small area imaging gamma camera, which was the outcome of the research work of Dr. Murli Ravi, and this is being now extended to a system which could be an handheld system roughly of the diameter of around 7 centimeters. This is a nearly real-time gamma infrared optical handheld intraoperative probe. That's why it's called multimodality, because it involves not only just detection of gamma radiation, but it also is going to have sensors that could detect infrared and optical radiations. The existing state-of-the-art probes are usually based on gamma Geiger counters, but this probe is the first imaging gamma intraoperative probe. This will ensure a drastic reduction in post-operative residual diseases, patient-surgeon radiation exposure, as well as ease of operation. And our aim is to have it as a very small, compact, handheld device that could be of great help to the doctor inside the operation theater. With minimal working distance of less than a centimeter, this probe is capable of real-time imaging, combining the optical IR as well as gamma images. Another area of work is on novel nuclear medicine imaging modality, wherein we are looking at introducing and establishing a novel nuclear medical imaging modality which addresses the weaknesses of PET and SPECT. Our work aims at has been on designing an optimization of a collimator for a new PET system using gate simulation. We have developed reconstruction algorithms, and this particular modality is a marriage of collimators of SPECT and time coincidence PET. The new candidate isotope which is being researched is the potassium 43, which has a longer half-life as compared to the existing use of isotopes which have very short-lived. This is a collaborative work which involves 6 institutions including SSSIHL across the globe, and the fabrication of this and testing of this particular instrument micro-PET will be at Riken, Japan. There are other exciting areas of research which is going on, which are listed here. The electronic structure theory calculations, nuclear structure studies in collaboration with the UGCDA consortium, where we'll be using the accelerator facilities, novel materials for capacitors and varistor applications, multifunctional rare earth materials for magnetic properties, and metal nanocrystal embedded classes for optoelectronic and photonic applications. I would like to draw your attention on the last 3, which are very interesting work. The Raman Spectroscopy, as all of us know, is a very good tool, and here we are using Raman Spectroscopy as a tool for medical diagnostics, which involves non-invasive probe and in-depth clinical diagnostics, which could be used for profiling bones. Already a lot of work has been carried out in this area, and we are continuing on this. We have proposed a collaborative research with Professor Antonio Preferi from the Politecnico di Milano, Italy. The other area is the other important work which is being carried out by a team is the development of biomaterials for medical applications, which focuses on better dental implant systems. We have signed an MOU with Professor Håkan Engqvist, Uppsala University, Sweden, and the team has been working on novel materials, biomaterials. And the last one, but not the least, is in the area of optical networking, which is optimization of DWDM networks and C+L band engineering, wherein we are looking at the optimization of a dense wavelength division multiplexing network by using a proper routing and wavelength algorithms and optimal regenerator placements. And we are also studying the linear and nonlinear impairments in the C+L band fiber optic networks, which is the current research in the industry. This particular research work is industry mentored research and has a lot of relevance to current day optical communication networking industries like Tejas Networks, Sienna, Cisco, Infinera, etc., where many of our MTech students have gone in as employees. Of course, no doubt, all this research work has been possible with the blessings of Bhagavan Sri Satya Sai Baba, and also the fact that we have got very good funding from external agencies and good support from the institute administration. We have collaboration with many institutions across the globe, and we also have interdepartmental collaborations within the institute. Thank you. Jai Sai Ram.
Thank you, sirs, for the informative and inspiring sessions on the research work that is being carried out in the various science departments. I'm sure student brothers will be really inspired to take this research further and get good laurels to the institute. We now have a session on the results for the various competitions held as part of NSD 2022. Over to the coordinators to take this session further. Thank you. Saira.
Om Shri Sai Ram. My humble pranams at the lotus feet of our most beloved Bhagwan. Respected elders, guest speakers, teachers, students, Sai Ram to one and all. I heartily welcome you all to the penultimate proceeding of the morning session of this year's National Science Day celebration. It gives me immense pleasure to host this exciting session, that is the announcement of results for various competitions held as part of the National Science Day celebration 2022. As you are all aware that every year we conduct various competitions for all the science enthusiasts to enhance their scientific zeal. This year also, we have had competitions like sketching, painting, essay writing competitions, and poster presentation. We have held competitions for the postgraduate students, undergraduate students, students from the higher secondary school, and also schools in and around Puttaparthi. Now I take this opportunity to announce the prize winners from the Sri Sathya Sai Higher Secondary School students from class 3 to class 12, both the boys and girls students. So we have, as I mentioned, we have had the sketching and painting competitions. So these were the few of the topics which we have given them. Firstly, the renewable energy, time to conserve it. Second, keep our air clean. Third, effective waste disposal and management, creating a better tomorrow. Fourth one, temples of healing. And fifth one, the science meets spirituality. In the essay writing competitions, the topics which were given were science, a blessing for mankind; climate change, a great threat to our existence; societal changes and lessons learned from the current pandemic; fourth, India, the emerging superpower; and last, temples of learning. So coming to the first section, that is, I'll be telling you about the juniors and senior section, boys section. Firstly, the sketching and painting competition. I would like to tell you all, please give them a big round of applause wherever you're seated. So the first prize in the sketching and painting competition for the juniors and seniors. So first prize goes to Sai Anupam Sahu of 10th class. Second prize, V. Umesh, 10th class. Third prize goes to DS Amaganata Sai Rama, 10th class. And consolation prize is backed by YT Vinay Kumar of 10th class. Coming to the senior section, first prize goes to P. Praneeth Reddy, 12th class. Second, Aditya Kiran Devan, 12th class. Third prize goes to Bharadwaj S of 12th class. Coming to the sub-juniors and junior section, under the painting and sketching competition, first prize goes to Venkatasai Vikshan Chitakonda, 5th class. Second prize goes to Tirumala Reddy, 4th class. Third prize goes to Sai Suhrit of 3rd class, and consolation prize goes to Pranav of 3rd class. And coming to the junior section, first prize goes to Aditya Batra of 7th class. Second prize goes to Sai Nandan, 6th class. Third prize goes to Mohit of 6th class, and consolation prize goes to Prajwal of 7th class. Coming to the essay writing competition under the juniors and senior section, boys section, that is, first prize goes to Pratyusha Sahu, 10th class. Second prize, K. Sai Chetan, 10th class. Third prize goes to Surya A, 10th class, and consolation prize goes to Ram Singh of 10th class. Coming to the senior section, first prize goes to Soham Kaushal of 12th class. Second prize goes to Sai Siddhant Botta, 12th class. Third prize goes to Naman Ahuja of 12th class. And consolation prize goes to N. Shravan Sai, 12th class. Coming to the sub-juniors and junior section, essay writing competition. First prize goes to Tej Reddy, 5th class. Second prize goes to Bhaskar Rao of 4th class. Third prize goes to S. Raju of 4th class, and consolation prize goes to Lakshman of 3rd class. Coming to the junior section, first prize goes to Anoop, 7th class. Second prize goes to Atharva, 7th class. Third prize goes to Sai Shreyas, 6th class, and consolation prize is backed by Y. Jagdeep of 6th class. Coming to the girls section, senior section, that is under the sketching and painting competition, first prize goes to N. Haripriya, 12th class. Second prize goes to P. Priya Reddy, 11th class. Third prize goes to Sharanya, 10th class, and consolation prize is backed by K. Pujita of 9th class. Coming to the juniors and sub-juniors and junior section, under the sketching and painting competition, first prize goes to Kartika, 5th class. Second prize goes to Mrinmai, 4th class. Third prize goes to Trushar Gajjar, 3rd class, and consolation prize goes to Vishnupriya of 4th class. Coming to the junior section, first prize goes to Tejasvi of 5th class. Second prize goes to Yashika of 6th class. Third prize goes to Sambhavana Reddy of 6th class, and consolation prize is backed by Jayapriya of 7th class. Coming to the senior section for the essay writing competition, first prize goes to Harshita Nagpal, 12th class. Second prize goes to Varshini Shri, 11th class. Third prize goes to Divya Ramji of 10th class, and consolation prize is backed by Anvita Vadlani of 9th class. Coming to the sub-juniors and junior section, essay writing competition, first prize goes to Kriti Sai, 5th class. Second prize goes to Akshara Agrawal, 5th class. Third prize goes to Sai Sumiran of 4th class, and consolation prize to Pujasvi of 3rd class. And coming to the junior section, first prize goes to Sai Gayatri of 8th class. Second prize goes to Sai Sashrita, 8th class. Third prize goes to Jayapriya of 7th class, and consolation prize is backed by Disha of 6th class. Thank you. Now we have a few of the painting competitions for the benefit of all. Like these are the few of the painting competitions that have come out from our higher secondary school students. So it is said that participation is better than winning, because if you don't participate, then there is no chance of winning. So I heartily congratulate all the participants for their wholehearted involvement in these competitions. Thank you, Sairam.
Om Sri Sairam, offering my humble pranams at the lotus feet of Bhagavan Sri Sathya Sai Baba. I take this opportunity to announce the prize winners of the various competitions held for our 3 campuses: Prasanthi Nilayam campus, Vrindavan campus, Mudanahalli campus, and also outside schools. So first of all, I would like to congratulate all the participants, because participation is the first step before anything you can achieve. So I congratulate, and various topics were chosen to conduct these prizes. So the topics were in line with the National Science Day theme given by the Government of India. So we have conducted essay writing and poster presentation for the UG and PG boys, postgraduate boys. So the essay writing competition, the UG PSN campus, the first prize goes to Manan Luthra, second BSc MES group. And poster presentation, the first prize goes to Manan Luthra again, second BSc MES. Third prize winner is Damodar Reddy K, first BSc MES, and third prize winner is Sai Sharat Aditya, first BSc MES. Now PSN campus postgraduate students winners, essay writing competition from the Department of Chemistry, PSN campus. First prize goes to Chinta Jagadish, first MSc. Second prize goes to Sri Mainak Mukherjee, first MSc. And third prize winner is Sri Uday Sai Reddy, first MSc. In the poster presentation competitions, first prize goes to Sri Shivanish Tiwari, second MSc. Second prize goes to Sai Atmiya, second MSc. Third prize winner is Varakrishna Mumidi Varapu, second MSc. And consolation prize in this category goes to Arpit Aswati, second MSc. D Physics, from the Department of Physics, PSN campus, boys, under the poster presentation competition, first prize goes to Jeevan Reddy and Katakam Devharsha Sivasai. Second prize winner is Mr. M. Akil, second MSc. From the Mudanahalli campus, first prize in the essay writing competitions, first prize winner is B.R. Sri Charan, third BSc Computer Science. Sri Sai, second prize winner is Sai Satvik, third BSc Computer Science. Under the poster presentation competitions, first prize winner is Reshav Nandlal, third BSc Computer Science. Tilak, second prize goes to Tilak D, second BSc Computer Science. And third prize goes to B.R. Sri Charan, third BSc Computer Science. From the Brindavan campus, under the essay writing competitions, first prize goes to Aditya DSS, third BSc Mathematics Honors. Second prize goes to Gandoli Sarath, second BSc MPC Group. And third prize winner is Ishan Bhatnagar, first BSc MPC Group. Under the poster presentation competitions, first prize goes to Sai Sanjay, first BSc MPC. And second prize winner is Arun Sai, second BSc MPC. And third prize winner is Deepanshu Patnaik, first BSc MPC Group. Once again, I congratulate all the participants and the winners of the competitions. Now, I take this opportunity to announce the prize winners. From the different schools. This is inter-school competitions that we have conducted. Again, in the sketching and poster competitions, sketching, painting competition and essay writing competitions. So themes were again in line with the Government of India announced theme for the National Science Day. And again, also the socially relevant and current challenges. So under the painting and sketching competitions, under the junior girls category, first prize goes to B. Aishwarya, 7th class, Sri Sai Vidyaniketan School, Puttaparthi. And second prize won by J. Tanusri, 8th class, SSS English Medium School, Puttaparthi. Again, the third prize won by Vaishnavi, 7th class, by Vasavi English Medium School. Senior girls, the first prize goes to S. Farjana, 10th class, Sri Sai Vidyaniketan School. And second prize goes to C. Deepika, 9th class, JPH School, Puttaparthi. And third prize goes to G. Kirti, 9th class, Vasavi English Medium School. And also again, one more third prize goes to Miracle Moga, 9th class, Sai Gurupada School. Now boys, under the painting and sketching competitions, junior boys, first prize goes to K. Manoj Kumar, 8th class, SSS English Medium School. Second prize winner is N. Nitvik Sai, 8th class, Sri Sai Vidyaniketan School. And third prize winner is V. Pawan Kumar, 8th class, Vasavi English Medium School. Senior boys, painting and sketching competition, first prize goes to S. Chaitanya Sai, 10th class, JPS School, Puttaparthi. And M. Nitish Kumar, 10th class, second prize goes to M. Nitish Kumar, 10th class, SSS English Medium School. Third prize winner is K. Tammai Srivasav, 9th class, Sri Sai Vidyaniketan School. Under the essay writing competitions, girls category. First prize goes to D. Naushin, 9th class, Sri Sai Vidyaniketan School. Second prize goes to Jyotirmai, 8th class, Sai Gurupada English Medium School. And second prize goes to, again, 8th class, Sri Sai Vidyaniketan School. Under this essay writing boys juniors category, first prize goes to T. Ashit, 8th class, Sri Sai Vidyaniketan School. Second prize winner is S. Nikileshwar Reddy, 7th class, Gurupada School. And third prize goes to K. Tarun Teja, 8th class, SSS English Medium School. Boys seniors winners under the essay writing competitions. The first prize winner is T. Venkat Bhargav, 9th class, Sri Sai Vidyaniketan English Medium School. Second prize goes to S. Vinay, 10th class, SSS English Medium School. And third prize winner is C. Tejas Krishna, 10th class, Vasavi English Medium School. Thank you. Once again, I congratulate all the participants and winners of these competitions who have taken the enthusiasm and showed the spirit of the science, and these children are the futures of the Indian science and technology. Thank you once of all to all the participants. Sairam.
Thank you, sirs, for the results. We now pray to Bhagavan Sri Satsai Varu for his divine discourse. Thank you.
ఈ ఆధునిక కాలమునందు అట్టి ఋషులు కలరా అని మనకు సందేహం కలగవచ్చు.
Today we might be assailed by a doubt, do we have such sages in our midst today?
ఈనాడు విజ్ఞాన శాస్త్రము కేవలము గొప్పది కాదని చెప్పకూడదు.
Now you cannot possibly say that what you have gained in terms of science and technology is not a great deal.
విజ్ఞాన శాస్త్రము అత్యవసరమే.
Certainly you do need the benefits of science.
అయితే విజ్ఞానమును అభివృద్ధి పరచుకుంట ఎంత అవసరమో, అభివృద్ధి పరచుకున్నటువంటి విజ్ఞానమును సద్వినియోగం చేసుకునేటువంటి వివేకం కూడాను అంత అవసరమని గుర్తించాలి.
Even as you have to further the cause of science, you should also understand to know how to make use of this scientific knowledge.
విజ్ఞానం ఉండి వివేకం లేకపోవటం చేతనే ఇట్టి విజ్ఞానం పెడమార్గం బట్టి దేశమునకు అనేక విధములైనటువంటి బాధలను భయమును కలిగించుతున్నవి.
Today you have the growth of science, but you do not have the discrimination how to use this advances in science. That is why the world at large is beset with
ఈ
problems.
ఆధునిక విజ్ఞాన శాస్త్రములందు కూడాను ఆధ్యాత్మిక తత్వాన్ని ఇమిడ్చి కొంతవరకు నువ్వు గుర్తించి పరిశోధనలు సలిపి పరిశీలనలో మానసిక పరివర్తన గావించినటువంటి మహనీయులు కూడా లేకపోలేదు.
Even in these days of this advance in modern science, there are some great many people who have understood the basic synthesis between modern science on the one hand and spirituality and ancient wisdom on the other.
న్యూటన్ భూమికి ఆకర్షణ శక్తి కలదని గుర్తించి తదుపరి తానే యోచించుకున్నాడు. నేను ఈనాడు దీని ఆకర్షణ శక్తిని గుర్తించినాను కానీ నేను గుర్తించక పూర్వం కూడాను దీనికి ఈ ఆకర్షణ శక్తి ఉన్నది అని తలంచినాడు.
Newton was the one who was the first one to have given an interpretation regarding the force of gravity which the earth has. But he also had the wisdom to know that this force of gravity, this force of attraction has been present all along.
అతను కూడాను ఒకానొక సమయంలో ఈ యొక్క ఆకర్షణ శక్తిని నేను గుర్తించాను కానీ సృష్టించలేదు అన్నాడు.
In fact, he went on to say that I have discovered this idea of gravity, but I have not created it.
దీనికి ఒక సృష్టికర్త ఉన్నాడు అని కూడాను విశ్వసించాడు.
He also had this firm conviction that there is that supreme hand behind this.
ఐన్స్టీన్ కూడాను ఈ యొక్క ప్రకృతి యొక్క శక్తి సామర్థ్యములను తన పరిశోధనల ద్వారా గుర్తించి తదుపరి ఒక ఆధ్యాత్మిక మార్గంలో ప్రవేశించాడు.
Einstein was yet another towering figure who delved deep into the mysteries of nature, and he found in the end that there is this idea of spirituality deep within all that is material.
ఈ యొక్క దివ్యమైనటువంటి ఆధ్యాత్మిక మార్గంలో ప్రవేశించవలనంటే సత్సంగం అత్యవసరమని కూడాను, సత్సంగం ద్వారానే మానవుని యొక్క మానసిక పరిణామం కూడాను జరుగుతుందని కూడాను గుర్తించాడు.
He further understood that if you wish to make the best of your knowledge pertaining to the nature, you need to have this satsanga, because it's only then that your mind and heart could be cleansed and purified.
అప్పుడే తనకు తాను ప్రశ్నించుకున్నాడు. టెల్ మీ యువర్ కంపెనీ, ఐ షల్ టెల్ యూ వాట్ యు ఆర్ అన్నాడు.
It was in this context that he once said, tell me your company, I shall tell you what you are.
మన యొక్క సంఘం ఎలాంటిదో, మనము అలాంటి వారము అనేటువంటిది ఈ ప్రశ్న వల్ల స్పష్టమవుతూ వచ్చింది.
What becomes manifest in this simple question is, the type of society we live in is a reflection of our own nature.
హైజెన్బర్గ్ అనేటువంటి ఒక మహాతత్వవేత్త,
Then we come to yet another great personality in the form of Heisenberg.
ఇతను అనేక వైజ్ఞానిక శాస్త్రములను పరిశోధన చేసి తదుపరి ఆధ్యాత్మికములకు వైజ్ఞానమునకు ఉండినటువంటి సన్నిహిత సంబంధ బాంధవ్యం ఏమిటని విచారించడానికి ప్రయత్నించాడు.
He made an attempt. He was one of those who contributed a great deal to modern thought and modern advances in science. Nevertheless, he too wanted to know this essential link between science on the one hand and spirituality on the other.
అప్పుడు తనలో తాను ఈ యోగ శాస్త్రంలో ఉండినటువంటి రహస్యాన్ని గుర్తించాడు.
It was in this event.
మాస్టర్ ది మైండ్, బి ఏ మాస్టర్ మైండ్ అన్నాడు.
He summed up his experiences in this manner. Master the mind and be a master mind.
హైడ్రోజన్ అనేటువంటి ఒక తత్వంలో ఎన్ని విధములైనటువంటి పరిశోధనలో సలిపి తదుపరి దీనికి ఏదో ఒక ముఖ్యమైనటువంటి ఆధారం ఉన్నదని ఒక విజ్ఞానవేత్త గుర్తించడానికి ప్రయత్నించాడు.
Now it so happened that one scientist wanted to know the nature of hydrogen, and he also wanted to know what the basis for this hydrogen was.
అతనే ష్రోడింగర్.
The name of this person is Schrödinger.
అతను ఈ వైజ్ఞానమునకు ఆధ్యాత్మికమునకు అంతర్ సంబంధం కలదనియు, వైజ్ఞానమునకు ఆధ్యాత్మికమే ఆధారమనియు నిరూపిస్తూ వచ్చాడు.
He went on to understand that there is a synthesis between science and spirituality, and in fact it is spirituality which is the basis of science.
ఇందువలన కోరికలను అధికముగా పెంచుకోవటం చేత మానవుని యొక్క తెలివితేటలు క్రమక్రమేణా పల్చబడుతున్నవని కూడా తాను గుర్తించాడు.
He also understood that by increasing one's wants and desires, one tends to lose one's own innate capacity.
అందువలననే మోర్ డిజైర్ ఇస్ మోర్ డిస్పైర్ అన్నాడు.
That's why he said, more desire is more despair.
తదుపరి డెరాక్ అనేటువంటి మహాతత్వవేత్త ఉండేటువంటి వాడు.
Then we come to yet another person, a great scientist in his own right.
ఇతను అనేక విధములుగా కూడాను ఆధ్యాత్మిక తత్వాన్ని అభివృద్ధి గావించుకొని తద్వారా ఈ యొక్క విజ్ఞానమునకు ఆధ్యాత్మికమునకు ఉండిన సంబంధం ఏమిటి అని కూడాను గుర్తించడానికి ప్రయత్నించాడు.
He also strived to seek this harmony between science and spirituality.
ఈ విధంగా సర్వజీవుల యందు ఉండినటువంటి యొక్క దైవత్వాన్ని గుర్తించి ఈశ్వర సర్వభూతానాం అనేటువంటి పదమునకు చక్కని నిరూపణ చేయడానికి శోధన చేస్తూ వచ్చాడు.
And what did he do? He tried to seek the one entity which lives in all living beings, in all living forms.
ఆ యొక్క పరిశోధన సలిపి లవ్ ఎవర్ హర్ట్ నెవర్ అనేటువంటి యొక్క సత్యాన్ని గుర్తించూ వచ్చాడు.
As a result of this pursuit of his, he came to the conclusion, love ever, hurt never.
అష్టాదశ పురాణేషు వ్యాసస్య వచనద్వయం పరోపకార పుణ్యాయ పాపాయ పరపీడనం అనేటువంటి వ్యాసుని యొక్క 18 రచనలందు ఉన్నటువంటి యొక్క సారాంశాన్ని ఈ డెరాక్ అనేటువంటి యొక్క సైంటిస్టు చక్కగా 2 పదములలో తాను నిరూపిస్తూ వచ్చాడు.
This modern scientist Dirac, what he had come to the conclusion is nothing but an echo from what the ancient sage Vyasa had given to mankind as the quintessence of all the Puranas, that never do any harm to others, love everyone.
డిప్రోగ్జీ అనేటువంటి ఒక మహా తత్వవేత్త ఉండేటువంటి వాడు.
And then we have yet another giant figure of science, de Broglie.
ఇతను అనేక విధములుగా కూడాను ఆధ్యాత్మిక తత్వాన్ని విమర్శించి తదుపరి నా విమర్శ కేవలం తెలివి తక్కువ విమర్శ అనేటువంటి యొక్క ఉద్దేశాన్ని అభివృద్ధి పరుచుకున్నాడు.
He began by trying to look for shortcomings and pitfalls in this idea of spirituality. He started criticizing spirituality.
దైవత్వం అనేటువంటి యొక్క శక్తి సర్వత్రా వ్యాపించి ఉండదనేటువంటి యొక్క సత్యాన్ని కూడాను తాను అనుభవరూపకంగా నిరూపిస్తూ వచ్చాడు.
But in the end, he came to the conclusion that this has been a futile exercise on his part, and he came to accept the existence of God, and that God is indeed behind every act.
వైజ్ఞానిక శాస్త్రమునందు కూడాను ఇలాంటి మహర్షులు లేకపోలేదు.
Therefore, the sum and substance is that even in these days of modern science, we have these present-day rishis.
వైజ్ఞానిక శాస్త్రమునందు ప్రకాశించినటువంటి, ప్రబోధించినటువంటి, పరిశీలించినటువంటి, పరిశోధించినటువంటి మహాభక్తులు ఈ సప్తర్షులు.
Some of these people are the present-day sages. In the same manner, going back to ancient times, the 7 rishis, the Sapta rishis, were those who gave us to us pearls of wisdom.
Newton.
Now the present-day Sapta rishis are Newton.
తదుపరి ఈ యొక్క సత్యాన్ని గుర్తించి ఈ జగత్తంతా కూడాను దైవం యొక్క నిర్మితమే, దీనికంటే గొప్పది కాదు, ఈ దైవ నిర్మితం చేతనే ఈ జగత్తు చుట్టుతూ ఉన్నది అనేటువంటి సత్యాన్ని ప్రపంచానికి చాటుతూ వచ్చాడు.
He gave to us the fundamental truth that it is God alone who has created this world. It is God alone who has given the properties that one associates with this earth.
తదుపరి క్రిస్ జాబ్ కాప్రా అనేటువంటి ఒక తత్వవేత్త అణువునకు తరంగములకు ఉన్నటువంటి సన్నిహిత సంబంధ బాంధవ్యాన్ని కూడాను విచారణ చేస్తూ వచ్చాడు.
There is yet another personage in this list, Fridtjof Capra, who unraveled this link between the atom on the one hand and the wave nature.
మహాభారతములో వ్యాసుడు రచించినటువంటి యొక్క తత్వాన్ని చక్కగా గుర్తించి స్థిత అనేటువంటి తంతువుల ద్వారా సర్వము కూడాను సృష్టింపబడి భువనములు భూతములు రెండు కూడాను దీని వలనే కలుగుచున్నవనేటువంటి సత్యాన్ని నిరూపించటం లోపల తరంగములు అణుశక్తి యొక్క సత్వాన్ని కూడాను తాను పోలిక చేసుకుంటూ వచ్చాడు.
Now in the ancient times, it was Vyasa who cognized the truth that in this form of this word Sita, which permeated everything and everywhere in the form of deep vibrations, which gave rise to all the worlds, all the elements.
We come to a close for the morning session. Thank you, sirs and brothers, for being a good audience for this morning session. We'll now break for lunch and reassemble at 2:55 p.m. sharp for the second session of NSD 2022. Sai Ram.